Welcome to Mr. D's class. This is a 3-minute bio on enzymes.
Okay. So what we're going to try to do is explain enzymes in under 3 minutes. Somebody have a timer?
I got it.
You got it?
56,
All right.
3 minutes right now.
Give me a countdown.
3, 2, 1, go.
Okay.
Slow.
Enzymes are a really special class of proteins. So proteins have all different functions, and enzymes specifically are the proteins that are going to help make or break covalent bonds in the human body. As a matter of fact, very few to none, or very few, let's just say, reactions happen in the human body without the presence of an enzyme. So enzymes are essentially strings, long strings of amino acid. Those strings have charged parts, and those charged parts cause the string to fold in on itself. They fold in, and then they have these complex structures. Now, all enzymes have a couple things in common. First of all, they're all going to have an active site, and they're all going to move in some way. They need to move in order to break or make the covalent bond, and the place in which they're making or breaking the covalent bond is called the active site. Some of these enzymes have inhibitors. You can block the active site. We call that a competitive inhibitor. And some have allosteric inhibitors, which will cause a shape change in the enzyme, causing the enzyme to basically close off the active site or make the active site inaccessible. We call those allosteric inhibitors. There are other ways to destroy a protein. If you change the temperature too much from its set temperature, then it will either unravel if it becomes too hot, or if it becomes too cold, it will literally freeze, stop twitching, and stop making or breaking the enzyme or the substrate. And the other way to change an enzyme is by changing the positive and negatives in the environment. If you introduce a whole lot of positives or a whole lot of negatives, you're going to change the way that string folds, and changing that way the string folds is going to change the structure, which is going to change the function of the enzyme, and that's it. Time.
1 minute. You have 1 minute left. What's a substrate?
Hi.
A substrate is going to be the thing that we are adding the covalent bond to or breaking the covalent bond from. The examples we've used are alcohol, breaking into acetaldehyde. We've used starch, breaking into glucose, and then we're going to have MAC kinase, which is going to add a phosphate group or form a covalent bond between a phosphate group and another protein. Other questions? 1 minute. 1 minute. Yes?
Can you describe some general characteristics of enzymes?
30
No.
seconds. 30 seconds.
Yes?
Do the inhibitors just come with the enzymes, or are they like outside of it?
They can be part of your body, so there are inhibitors for enzymes naturally that occur in your body, or you can add them in the case of antabuse that we had for alcohol, right?
I think time's up. Wait,
Yes.
just kidding.
You said like the chart is the environment. If they change the exterior of an enzyme, like what does that mean? What's an example of that?
So adding hydrochloric acid to an enzyme that works in your brain is probably going to screw it up.